Hi, so we just got done talking about the innate immune system. Now we're going to go on to the other branch, the adaptive immune system. And if your body's a castle, I want you to think of the adaptive immune system as the actual people there manning the castle, the guards, all the staff that are on hand. Our adaptive immune system is slower than the innate, but it is highly specific toward a particular pathogen. Sometimes it's also called the acquired immunity, and the key feature is that this system can remember past infections. So on this graph, if we've got on the y-axis number of antibodies being made, we can use this as a measure of how strong or how robust the body is at reacting to a particular pathogen. Or in this case, it's called an antigen. At the first exposure, first time your body is exposed to this antigen, it creates some antibodies slowly, and then as the disease is fought off and you recover, the number of antibodies goes down. However, the adaptive immune system remembers this antigen, and when that same one comes in your body a second time, the response is now not only far greater, more antibodies are made, but also the slope of this line is steeper, so it's making more antibodies faster than it was the first time. This difference between primary response and secondary response is a hallmark of adaptive immunity. So first we're going to talk about how the adaptive immune system can be so specific. We have cells that respond specifically to every single possible pathogen we can encounter. This specificity is achieved through the antigen-antibody binding. These antigens are foreign molecules, and there might be, say, a cell with a bunch of different antigens on its surface, but what's important is that the body can recognize any foreign cell or virus that invades it. Our body produces these proteins called antibodies. Our body produces antibodies, which have two arms, and on each arm is an antigen binding site that is specifically shaped. It's a protein that's specifically shaped to bind tightly, strongly, and specifically only to the antigen that it's made for. So in this case, the yellow antibody will only bind strongly to the yellow antigen. And your body makes antibodies that will bind to this purple one, to this red one, to this green one, all different antibodies you're capable of making. And it's this specific fit between antibody and antigen that allows our adaptive immune system to specifically attack each foreign pathogen. Antibodies can be free-floating like this one, or they can be attached to the surface of a cell called a lymphocyte. And in either case, they're doing the same thing. These arms are binding to the antigens, identifying the foreign molecules. Now what's interesting is that among antibodies, we make millions of different ones, and they come in several classes. There's one class of antibodies called immunoglobulin A, IgA, and this class of antibodies is actually secreted through breast milk in high concentrations. So this means that when a mother is breastfeeding, she's passing down antibodies, a form of immunity, to her child. And so that's actually one of the benefits of breastfeeding. Another class of antibodies called IgG is present and can cross the placenta during gestation. So in that way, a mother can transfer her immunity to the fetus before it's even born. So in this diagram, we've laid out the white blood cells, also called leukocytes, into the ones that are involved in innate immunity, and over here the ones that are involved in adaptive immunity. In adaptive immunity, the cells are called lymphocytes, and we divide that into T cells and B cells. Both of these lymphocytes are produced, they are initially born in the bone marrow. B cells, they will stay in the bone marrow and mature there. Here, this bone marrow is producing a B cell to detect every possible antigen that may be encountered. So that's what I mean by the B cell maturing. They're maturing to be specific toward every antigen that can be encountered. The T cells, however, they will start in the bone marrow and they will move into the thymus, which is this organ that we have down our chest here, and they mature in the thymus. As well, the T cells are maturing in order to recognize a specific pathogen. When these cells are mature, they're going to be released into both the blood and the lymph. We all know about the blood circulation, and the lymph circulation is another transport network throughout our body that works more in getting rid of the unwanted materials of the body, whereas the blood also works in delivering the materials that we do want, like delivering glucose and oxygen to all the parts of our body. Since lymphocytes are made in the bone marrow, people who have lymphoma, which is a type of cancer of the bone marrow, they can receive a treatment in the form of a bone marrow transplant. And when this is done, you just have to be very careful to make sure that the person donating the bone marrow is a match in terms of the types of receptors that are displayed on their cells, so that the bone marrow will be a match and the body won't try to destroy its own cells. So you may be wondering, how exactly can our body produce millions of different antibodies, one for each possible antigen that we might encounter? How do we possibly have enough space in our DNA to do that? Well, that is done through a process called recombination, and sometimes it's called VDJ recombination. Up top here, we're starting with the DNA of an undifferentiated B cell. So this is a B cell that hasn't yet gone through the maturation steps in order to be specific toward a particular antigen. Because remember, in the end, the B cell is going to have what's called here as a B cell receptor, but we can just call this an antibody on its surface. And so it's got these little Y shapes on its surface that are going around trying to recognize what is a foreign antigen. So we start with the DNA that's undifferentiated. There are several V segments, V for variable, some J segments, and C segments. The C segment is constant, so there's only one of those. Every antibody has the same C segment, which is this dark one down here. And the J segments join together, the constant and the variable regions. The variable regions, as you might expect, are the ones here. They need to change shape in order to bind to only their specific antigen. During maturation, the B cell will differentiate, and some parts of this DNA are going to be spliced out. Here in this one, all but 3 of the V segments have been spliced out, and 1 J segment is left over. Now I want to point out that this is not like mRNA splicing that you've learned about recently. This is different because it's happening in the DNA. Once these V segments get chopped out, they are gone forever in this B cell. In this case, they've kept V1, V2, and V3, but in another B cell, they may keep V11 and V12 and V13. So every B cell is going to be different. Once the B cell is mature, transcription can occur, producing the pre-mRNA. That's what we call it before it undergoes splicing. And then the mRNA once it's undergone splicing. The final mRNA should contain only 1 V, 1 J, and 1 C. So we've gone from many possible segments down to just one. If you consider that every B cell can undergo a slightly different combination of sections spliced out, you can see how the B cells can all end up with a slightly different specificity, different antibodies on the surface, and being able to recognize different antigens in the body. So we're going to talk about the B cells first. And in our castle story, these are the castle guards. They are constantly patrolling on guard. They circulate the blood and the lymph on guard for an antigen. We say that they are part of humoral immunity because the humors are an old word for the fluid of the body. So the humors include the blood, the lymph, the extracellular fluid, basically everything that's not inside a cell. The B cells are going to roam around the blood and the lymph, and they have those Y-shaped antibodies on their surface. Each B cell recognizes only its specific antigen. when a B cell finds its antigen, it's going to activate. And an activate B cell does two things. It undergoes a lot of mitosis. We call this clonal expansion, producing a lot more B cells. But the B cells it produces aren't going to be the same. Some will be plasma B cells, some will be memory B cells. So there's differentiation here. Here's our initial B cell, and it's been roaming around the body, and it detected an antigen, which bound to its antibody. When the B cell is activated, it does clonal expansion. Some of the cells that it divides into will be plasma B cells, and these are short-lived. They don't last for a long time, but while they are around, they just pump out hundreds and hundreds of free antibodies. And the value of this is that the free antibodies are going to be floating around everywhere. They're going to be able to attach to whatever was producing the antigen, the foreign material in the cell, and they're going to cover it up. Say if it's a bacteria, they're going to cover up the surface of the bacteria with all these free antibodies, and this will prevent the bacteria from undergoing its functions. Another good thing about having a lot of antibodies is that when a bacteria or another pathogen is covered in antibodies, this speeds up the speed at which it gets phagocytosed or eaten by the phagocytes. Plasma B cells only last long enough to fight the initial infection and then they die off. However, another class of B cells was produced called memory B cells, and they're long-lived. They're going to stay around in the cell even after this initial infection is over. And they're going to be around even when that same antigen happens to enter the body again. And this is important because when that same antigen enters the body again, now say there are a lot more B cells around to fight that infection. Say for the first exposure, there was only one B cell that responded to that antigen. At the next exposure, there are 4 because of these memory B cells that are long-lived. Now, I'm not saying that it's actually 1 becoming 4, but the number of B cells multiplies, and so the body is much better able to respond to the second infection. Here's a graph showing this. Here's the first antigen exposure, so the first time your body encounters this antigen, it produces some antibodies, but it's kind of slow and it doesn't make as many in total because there were not that many B cells around to respond. We produce plasma B cells, which secrete the antibodies and then die. And we produce memory B cells, which stay around long after. The body's exposed to the antigen again. This could be a year, 10 years down the road. And now, because these memory B cells that were made are still around, they're long-lived, the antibodies are produced at a faster rate and a higher maximum number of antibodies produced. The secondary immune response is always much stronger than the primary immune response because of those memory B cells. Now this is an important one. This diagram, primary versus secondary immune response, highly likely to come up on an AP exam as far as we know. So this is the one that you should remember. Join me in the next video when we look at T cells and their function in the adaptive immune system.